We let Greg pontificate a little bit longer there because he was giving us such good information as our prior speakers, but we're going to shift gears a little bit and ask Dr. Steve Jameson from the University of Minnesota to talk to us a little bit about the pet store mice and the dirty mouse and its effect on immunity and modeling as a change of gears. Thank you, Steve.
Is that on?
Yeah.
Can you hear me?
All right, if we put this up to the fire, it comes up better. There you go.
Thanks. Well, thanks to the organizers, thanks Joe and everyone else for the opportunity to see the changing of the battery. Fair enough, I'll do an interpretive dance until I can change slides. So I'm going to talk about a slightly different topic, and I've got a sneaking feeling I'm here as a kind of provocateur to challenge some of the widely held beliefs in this room. I'm not really trying to change everybody's perspective, but just to give you a different one. So what we've been doing in some studies that are collaboration between my lab and Dave Massapus is to try and ask this question of how immune system characteristics in mice would change if they had a more natural, more physiological immunological experience. So the immune system as a whole is really designed to respond to pathogens, to invasions. And my overall concern with more reductionist approaches where we're keeping mice separated away from those kind of normal, natural threats is that you have a situation where you've got an army, basically. I hate these militaristic analogies, but you've got an army prepared to respond to something and there's nothing challenging it. And does that create its own kind of level of noise? Okay. So what do I do? Okay, change slides, yeah, fine enough. So first off, I'd make the point that what we've learned from lab mice maintained in SPF facilities has been incredibly useful, groundbreaking work that's led to all kinds of important discoveries. I'm not going to go through this list, but nonetheless, there it is. And now, the big green one. Good-o. But I'm going to talk about something slightly different, where we've been working with what we're going to call dirty mice as a kind of shorthand. The question again is whether a broad, more physiological infectious history might alter the immune system and immunological responses in inbred mice. So trying to maintain the genetic component, something we can recognize, but change the immune experience of the animal. So this is a kind of overview. Our approach included analysis of wild-caught and pet shop or pet store purchased mice, which I'm going to call dirty mice, as well as co-housing pet store mice with inbred strains, so the latter will acquire some of the microbes transmitted from the pet store animals. So we know that these wild and pet store animals have been exposed to many pathogens, only some of which we can define, and with their own particular blend of commensal microbes. Some of these pathogens and commensals will be acquired by the co-housed mice, but we don't try to control this. We're letting things transmit by themselves, mouse to mouse, through the usual means. These are essentially the opposite of gnotobiotic mice. We don't know the infectious history of the animals, and that's definitely a limitation, but it can be seen as a more authentic reflection of human microbial experience, where we've all had different microbial experiences. So if we're trying to develop a model for human immunology, in my case, perhaps we should explore this. So there are huge logistical problems. These dirty mice are known to carry SPF-excluded pathogens, and perhaps ones we don't know so much about, which give the vets in our facility cold flashes. So an effective barrier with SPF colonies was absolutely required. We use BSL-3 housing for all this work, which is massively expensive, but really necessary because we don't really want to have any risk of any of these microbes leaping from our dirty mouse colony to a normal SPF colony. There's all sorts of ironies with this. These are mice we purchased from a pet store that my kids could buy and keep in their bedrooms, but we have to keep them in a BSL-3 facility next door to the TB lab. And anyway, so it's all fun. So this approach of deliberate sequential infection, which Skip has done, and I'm going to show stolen a couple of his slides, approach of deliberate sequential infection is a valuable alternative to this, where we can know what the infectious history of the animals is, but there is a problem in deciding which pathogens and commensals we should use for that deliberate infection or association. We started using microbes that frequently arise as chronic infections from the pet store animals and transmit to the lab strains. So we're trying to use a kind of blend of these things where we're allowing natural transmission of microbes that came from these pet store animals and were transmitted to the inbred strain mice, and say those are ones that really naturally jump pretty efficiently between animals, and start to work with those. There are some that we will deliberately include, such as MCMV, cytomegalovirus in mice, although actually we never find that in any of the pet store mice. So that, strangely enough, I thought it would be rampant, but it's never shown up so far. Okay, so I'm going to toggle back and forth between logistics and actual data just to explain where we've got with this project and what we want to do in theory. Yeah, so part of our driving force here was to say, you know, have we actually got a good system in mice for looking at human immune questions? Should be a little faster transition here. Nope, can't get it to work. Okay, thanks. These are just me, you keep going. Some reviews pointing out how the things we predict from mouse studies don't always apply to human studies. I probably don't have to convince you about that. But as a mouse immunologist, these are tremendously frustrating. We'd like to be able to improve on the models and be able to say that this is a more useful starting point for comparing the immune response in people. You or I get a vaccine compared to our lab mice. So part of the question is, what is a starting point? What kind of immune system do you have in a mouse versus a human before, let's say, vaccination or an infection? Next slide. This is some data that I think Andrew showed earlier, but this is from blood, in actual fact, from in the first panel there, B6 mice, a normal SPF mouse, and we're looking at CD8 T cells. Let's see, if you go forward, there we go, great. These are kind of color-coded just to save a little time. This phenotype of cells would be naive CD8 T cells. These would be the central memory and the effector memory populations. Most of the cells in a SPF mouse that hasn't been immunized with anything are naive, as one might have expected. Next. And actually, that corresponds pretty well with the situation in human cord blood. So from a neonatal human, different markers, which is part of the problem. But these are the kind of markers that people accept for designation of these same subsets. Sorry, a lot of clicks, I'm afraid. In humans, so again, naive population is about 80% in both. That's fine if we want to model human cord blood. But if we wanted to instead model the, adult humans, we're really not in the same ballpark here. So although there are still naive CD8 T cells in the blood of an adult human, there's a huge population of effector memory in this TEMRA population, which is really not reflected at all in the mice. And those cells do have characteristics such as high expression of granzyme B, a cytolytic component, that imply that they're kind of ready to kill should the need arise. So we think that just from this one subset in this one tissue, blood, we've really not got a very good representation. If we expand this a little and look in non-lymphoid tissue, Dave and I are both very interested in T cells that set up shop, become resident in non-lymphoid tissues. In a normal, healthy adult human cervix, there are some cells that are present. These are CD8 T cells within the parenchyma of the tissue, whereas in an SPF B6 mouse, nobody's home. There are no red dots, even though we're staining for them. There seems to be, until an immunization comes along, there's nothing present. Since these cells can shape both innate and adaptive responses that are induced in those tissues by infection, the presence of these cells, regardless of their specificity, might be important. So that's just enumerated there. Sorry. So there could be many reasons why the mouse and human immune systems would differ at this baseline measurement. Of course, 70 million years of evolution is not to be discounted. It's important. The animal lifespan and age analysis is important as well. Usually using mice within 2 years of life. That's pretty young for looking at humans. The one we can focus on, the one we can experimentally manipulate, is the immunological experience, the environment of the animals. And I'm just going to take a little diversion here that years ago there were discussions in the National Academy of Sciences talking about kind of initiation of germ-free and specific pathogen lab animals and how this is picking up in 1971. And this practice is gaining momentum. Most countries, despite the fact that some members of the older school of medical research consider that the results of tests carried out with the SPF animals may be misleading when extrapolated to man, who cannot be regarded as pathogen-free. So I guess I'm now old school, though I never thought I would be, but I'm having that same kind of question whether some of the SPF, let alone germ-free, but SPF studies may be a fantastic for reductionist approach, but may have its limitations. So don't attack all at once, but let's just consider the question. So, click. So mice used in, I don't need to tell you this, sorry. So using SPF conditions or germ-free conditions definitely helps normalize immune status prior to study and allows for reproducibility, but it artificially limits the normal physiological exposure of these animals to microbes. All right? So in particular, I'm thinking of pathogens. We could talk about commensals as well, and I do briefly, but let's think about just the onslaught of normal pathogens that we all get exposed to, myself included, and that would shape our immune system going forward. Humans, unlike these SPF animals, are exposed to all kinds of interesting things, some of them more serious than others. Keep going. These are all just gags here. Syphilis is always fun. So, so if the SPF mice are too clean, what do we see in dirty mice? And what do I mean by a dirty mouse in particular? Sorry. This is kind of what I showed you before. These are CD8 T cells. Just we could have picked any population, really, I think, but CD8 T cells happen to be what I like to work on. So from the blood of a lab mouse, again, most of the cells have a naive phenotype. If we look at them, if we catch wild mice and look at the CD8 T cells in those animals, a lot more cells have this effector memory phenotype. Correspondingly, fewer by percent have a naive phenotype. That would be an interesting thing to continue to work with. Is there are limitations with these wild-caught mice. You have to catch them every time. There could be, depending on the season and so forth, very occasionally it gets cold in Minnesota. So depending on whether those mice happen to come from some of the frozen tundra or in the middle of the summer, that might change their immune status. Also, they bite you when you catch them. So a lot easier to work with would be pet store mice, which we have here. And if anything, actually, the pet store mice just purchased at local Minneapolis and St. Paul, we tried to be even-handed, pet stores, have, if anything, a more severe phenotype, thank you, where there is a even higher frequency of effector memory phenotype cells from those animals. It blends out eventually if you do enough animals, but certainly the pet store mice are not lacking with this phenotypic conversion of the cells in the blood. Again, this is percentage. Absolute numbers of naive cells actually haven't really changed very much. Sorry. Thank you. So that led to the potential for some fairly straightforward experiments. We could do work with the pet store mice, but they're outbred, and we can do PCRs across highly polymorphic regions of the MHC and prove that to ourselves that they really are outbred. And we really wanted to work with inbred mice so that we can have some kind of control over this situation. So one approach is to do simple co-housing of the animals. Thank you. So if we take adult pet store mice, mix in some adult B6 mice, and then just wait. I'm not going to do this in real time, but we wait 2 months or so to see what happens, just allowing transmission between the animals. And again, this all needs to be done under BSL-3 conditions to avoid colony contamination. I don't want Mark Jenkins to lynch me. So what happens when we do that, and how do we monitor the transmission? So we use a fairly regular SPF panel serology for the most part to identify what these animals had. So these this is from a few years ago, but this is 15 pet store animals looking at this by serology of what they had been at some point infected with. So a lot of them with the red dots were infected with mouse hepatitis virus, norovirus, Sendai, Tyler's virus. A number of them had Mycoplasma pulmonis. All of them had pinworms and mites, the poor little things. So as you can imagine, this, for the vets in our place, they've never seen a scorecard like this. It's really, for a moment, it's really fun, and then they kind of walk away, wash their hands a number of times. And, again, BSL-3, okay? So that's what these mice had at some point been exposed to. It doesn't mean that they're carrying any of these things and they would transmit them. So an important thing was, by the way, our SPF colony is good. We got 0 for all of these from our lab mice. But if we co-house lab mice and pet store mice, do any of these jump? Sorry, next chunk there. Yes, some of them do anyway. MHV, I mean, a number of these you might have predicted. MHV is a good one at spreading. Sendai virus, Tyler's virus, both move pretty well. Mycoplasma pulmonis. Again, it's never 100%. We've never had really 100% of anything except pinworms and mites. So a number of these infections will transmit within one cage. If there are 3 lab mice, maybe one of them will get Mycoplasma pulmonis. So we're launching into this great area of variability, right? And we're kind of, at one point, embracing that and saying that's like this human population here. We've all had different immune experiences, but are there some common themes that would apply regardless of the particular immune experience you've had, okay? If you're looking for antibodies to, a Sendai virus, well, sure, it's only going to be animals that got exposed to Sendai virus that will score. But if we're looking for some more generic features of the immune system, would some of them have changed? And more importantly, would some of them be more in keeping with humans, and for which we're trying to model? Yep, so some microbes move. Sorry, there we go. So what happens when we do this co-housing? I've mentioned there's these microbes that move. Does anything happen in the circulating CD8 T cells? Yes, it does. So within a month of co-housing, a lot of this you might expect, right? These mice have now got infected with various nasty things that for years we've kept out of SPF colonies. This is part of the reason, because you provoke a very strong immune response. So within a month or so of co-housing, there's a very clear immune response by CD8 T cells that settles down by about 2 months, which is why we picked that time point, because we've, I might have taken this slide out, but basically the phenotype in the blood CD8 T cells stabilizes. Another consequence of this, thanks, is that putting these B6 mice in with pet store mice, some of the B6 mice will die. I might have, if I had been a betting man, I might have thought that this would happen early on as they first get infested with these various microbes that the pet store mice are carrying. That's not the case. So mostly this was a wasting disease that showed up fairly late. So by about 2 months is when it reached its limits, but about 20% of the mice died. We think this, the best association we've got so far is with Mycoplasma pulmonis, but not to be proven. So we do see now cells after co-housing, we do see cells showing up in non-lymphoid tissue. This is just, again, to highlight the fact that we're seeing changes in multiple sites. Keep going. Keep going. I threw this one in just to say that we have started to look, we haven't got very far into this, at the intestinal microbiota. I'm just going to mention that by diversity indexes, we really didn't see a huge difference between co-housed, pet store, and SPF animals. Go on. But if we look at the composition by PCA analysis, the microbiota, the SPF animals starts out in a very different place than the pet store animals, and after co-housing, it seems like the pet store microbiota tends to dominate. I'm not sure what that means, but it does imply that not only are we changing the infectious agents that we would typically count as SPF pathogens, but we're also changing the microbiota in these mice, significance to be determined. Can you keep, thank you. Okay, a major issue for the animal welfare protecting SPF animals is to keep the bugs where they're meant to be, so we're avoiding the spreading of these pathogens to our standard SPF colonies. We don't have, segregated employees who only work with the dirty mice, so we use the BSL-3 facility, as I mentioned, for this kind of study. We do have this incidence of some mortality in the co-housed animals. B6 mice are relatively tough. We get a higher mortality from initial studies with bulb C strains, which is proving to be a problem. Keep going. So we thought that SPF-designated pathogens would be the most likely cause for these deaths. So we're using necropsy and iterative processes to determine what pathogens get transmitted that cause that mortality. I mentioned Mycoplasma pulmonis. Click ahead, thank you. And using contaminated bedding instead of co-housing has led to some reduction in mortality without apparently compromising immune education. So we think that spread by fomites probably works to a fairly good extent, but may not be, obviously may not be exactly the same. One more. Sequential infection models, if they recapitulate the dirty mouse phenotype in terms of this basal immune status and immune activity, could help avoid this issue, though I think we'll never quite get past this point, even though we're using a lot of, we're thinking of using a lot of BSL-2 designated pathogens. Once you say things like MHV and Sendai and Mycoplasma pulmonis, the vets are not really willing to talk about BSL-2. We're already in a BSL-3 facility, and that's where we will stay. I don't really have time to go through this, but I just mentioned that when we have tried to ask this question of whether now with this co-housing of pet store animals or looking at pet store animals versus lab mice, do we actually get anything that's more similar to the human immune system? This was some gene expression analysis comparing the gene expression we see in these pairs of mice compared to adult and neonatal human blood. And there's a very good concordance. I'm going to skip through these because I'm running out of time. Punchline, whoops, let's go back one just briefly. Punchline would be that one of the major signatures, I haven't got time to explain this, but one of the major signatures we see that's conserved in these immune-educated mice and immune-educated humans, i.e. you and me, is a strong elevation of type 1 interferon induced genes. And so we think that that is a kind of preserved feature that matches between humans and mice. Keep going. This is stolen from one of Skip's papers where they were using deliberate infections. One more. And one more. Or, yeah, until it goes. And basically seeing a similar kind of pattern that, again, the effect of immunological experience, in this case with 3 viruses and a worm, would give you some similar trends of, some similar patterns of concordance between adult and neonatal human. Keep going. Okay. So there's some attention now that this study's out. I'm just going to say we're collaborating with PIs of various institutions as well as some small and large drug companies who want to look at these kind of animals in order to test their drugs, their approaches in animals that might have a more immunologically broad experience. Our SOPs still rely on BSL-3 containment. I'd love to have some other way of doing that. It would save me a lot of cash, but it's not actually required for any of the known pathogens carried by these mice. But I think anybody who would risk doing this outside of a BSL-3 facility may be asking for trouble. So let's stop there. It's a collaboration between my lab and Dave's. Many people involved. Click one more, especially Sarah Hamilton in my lab and Ali Bira in Dave's. Thanks very much.